Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this series of lecture on special products from amino acids. So here in this lecture, I will be going over details on glutathione, acetylcholine and biogenic amines. Let's get into each one of them. Glutathione, it is a tripeptide. That means there are 3 amino acids present in a glutathione. What are the 3 amino acids that are present in glutathione? One is a glutamic acid, other is cysteine, and the third is glycine in the sequence gamma glutamyl cystinyl glycine. And that's how the glutathione is present. Now this glutathione, it is an intracellular antioxidant and it's going to participate in the neutralization of certain reactive oxygen species molecules like the ones that are coming from hydrogen peroxide. So glutathione having a thiol group with a cysteine having SH group, and that H group can be donated in neutralizing a reactive oxygen species molecules. Now let's first see how a glutathione is synthesized, and then I will explain you how exactly it participates in neutralizing hydrogen peroxide so that hydroxyl radical or a free radical is not synthesized. So as it is shown here in the figure, cysteine is going to condense with glutamic acid to make gamma glutamyl cysteine. That is done by gamma glutamyl cysteine synthetase by using ATP. Now the gamma glutamyl cysteine, it is condensed with glycine by glutathione synthetase enzyme, which uses another ATP to make glutathione. And once glutathione is made, a sufficient glutathione, at that time glutathione will have a feedback negative effect on the first enzyme, that is gamma glutamyl cysteine synthetase. Now glutathione, it is written as GSH, and this GSH, say 2 molecules of GSH are used in the conversion of hydrogen peroxide into 2 water molecule. So hydrogen peroxide is neutralized into 2 water molecules so that hydrogen peroxide do not go into hydroxyl radical, because hydroxyl radical is a free radical. So that is prevented, right? So hydrogen peroxide is neutralized into water molecule by donating protons present in GSH. So these 2 GSH, now they will be converted into GSSG. And this GSSG, which is a oxidized glutathione, this is oxidized glutathione. So, and GSH is a reduced glutathione. So, oxidized glutathione upon neutralizing hydrogen peroxide, it will be converted back into GSH. Basically, it is reduced by enzyme glutathione reductase. So, glutathione reductase here, it is going to use NADPH plus H plus as the reductant and release that as NAD plus while NADP plus, while GSSG is converted to GSH. So this is how a concentration of antioxidant is maintained, especially by having sufficient NADPH plus, and that will come from pentose phosphate pathway, as we have seen this in pentose phosphate pathway lecture. Now the neutralization of hydrogen peroxide into water by using GSH, it will be done by enzyme glutathione peroxidase enzyme, glutathione peroxidase. So the glutathione peroxidase, it is a selenium containing enzyme. So this is some of the detail how a glutathione act as a major antioxidant in our cell. This is an intracellular antioxidant. Not only that, glutathione, it participates in transport of amino acids across the intestinal and renal tubular membrane coming from their lumen inside the cell. That particular transport of amino acids from the lumen of the intestine or lumen of the renal tubular membrane into the cells is done by gamma glutamyl cycle, and the gamma glutamyl cycle is in fact is the way to regenerate or to synthesize glutathione in our cells. Now coming to our next molecule is acetylcholine. Acetylcholine is a parasympathetic neurotransmitter and it is synthesized by combining or condensing choline coming from phosphatidylcholine with acetyl-CoA in the presynaptic neurons. Now how do we get that choline for acetylcholine synthesis? Choline will come from a molecule called phosphatidylcholine which is present in our cell membrane. In order to make phosphatidylcholine, so serine has to undergo decarboxylation to make ethanolamine first, and ethanolamine which is phosphorylated, so phosphatidylethanolamine is converted to phosphatidylcholine in the presence of three molecules, that is SAM, S-adenosylmethionine, folic acid or vitamin B12. So these are the three things that are necessary for the conversion of phosphatidylethanolamine into phosphatidylcholine, and that phosphatidylcholine, it is the most abundant membrane phospholipid. That means to maintain a good concentration or good amount of membrane phospholipid, that is phosphatidylcholine, we should have a sufficient S-adenosylmethionine, folate and vitamin B12. So that means in the deficiency of vitamin B12 or folic acid, there can be a decrease in the synthesis of phosphatidylcholine, and that may lead to membranal damage, and that might affect a synthesis of acetylcholine formation, which is a parasympathetic neurotransmitter. Now the figure here, it is showing you how a choline is combining with acetyl-CoA to make acetylcholine, and that is done by choline acetyl transferase enzyme. Now once acetylcholine participate in a neurotransmission process, so as with any other neurotransmitters, these neurotransmitters are short-lived, so they will undergo degradation, and the acetylcholine will undergo degradation by enzyme acetylcholinesterase into acetate and choline, and this is how acetylcholine will be degraded. So here the acetylcholinesterase enzyme, it is a serine esterase and it breaks acetylcholine, and there are certain drugs that can inhibit this enzyme which are referred as acetylcholinesterase. The examples physostigmine, neostigmine, and also certain like pesticides which belongs to organophosphate category like malathion, parathion, they all inhibit acetylcholinesterase and increases parasympathetic activities. That includes nerve gas like sarin, which are synthetic organophosphates that inhibit acetylcholinesterase. So overall, when acetylcholinesterase inhibitors are used, there will be increase in acetylcholine levels in the neuromuscular junction, and that basically increases the parasympathetic action. There is a disease which is related to this concept here called myasthenia gravis. Myasthenia gravis, it is an autoimmune neuromuscular disease where autoantibodies are synthesized against nicotinic receptors in the postsynaptic neurons at the neuromuscular junction. Thereby, these antibodies going to kill the nicotinic receptors. Thereby, availability or the presence of nicotinic receptors will significantly decrease. And this causes inhibition of excitatory effect of acetylcholine, leading to muscle weakness. Another treatment for myasthenia gravis is to use acetylcholinesterase inhibitors, thereby increase the concentration of acetylcholine in the neuromuscular junction, thereby some of the nicotinic receptors that are available there are continued to transmit the information, thereby improving the muscle contraction. Other ways of treating myasthenia gravis is to use immunosuppressants, and in selected cases thymectomy can be done. So, in myasthenia gravis, when we block acetylcholinesterase enzyme by using acetylcholinesterase inhibitors, so when you block here, so there is increase in acetylcholine, and that acetylcholine can bind to nicotinic receptor and improve the muscle function. That's what I was saying in the previous slide. So this is all about glutathione, acetylcholine and biogenic amines. Thank you.